Okay, good morning, everybody. My name is Sandara Brasil, and I'm presenting you a very cool project that includes the use of museum specimens with a genomic approach to understand how wild bees are responding genetically to land use change and to climate change. So it is very, very well known nowadays that biodiversity is declining. And the major threats, the biggest responsible for this are human land use and climate change. And this pattern, it seems to be happening to a lot of wild animals. And among wild animals, we have a very important pollinators that are bees. But what is still uncertain, it's at what scale this is happening, and also how specific taxa or species are responding to these changes, to their natural habitat. And a very complete and complex way to understand how these species are being affected or suffering from climate and land use change, and how this is really affecting the organisms, is to perform long-term studies. Although long-term studies are usually unfeasible because it requires a lot of money for research, a lot of people to do this research, sometimes both, and of course, a large amount of time. So to help cope with this, a approach emerges called Museomics. And Museomics use historic or ancient DNA from museum specimens and affords an opportunity to perform a time series analysis to spot genetic differences accumulated over decades for several species. And it is possible to assess phylogeny, pest pathogens, ancestral population structure, and also species response to global change. So here we use historic DNA and contemporary data to investigate genetic changes from two Ceratina species, the Ceratina coccolata and Ceratina dupla, to see how the populations are changing across a 50-year time span. So for this, we use two small capital bees, Ceratina species, the Ceratina coccolata and Ceratina dupla. Ceratina species are globally distributed. They have over 350 species, and they are the top 20 most they are among the top 20 most economically important bee species because of the higher value as a pollinator for both wild plants and crop species. So for this, we used 30 females individuals. We collected specimens from museum from 1968 and also collect the individuals from 2018 from the same sample spot. And we have 15 for each species. And we extract the DNA. We perform a RAD6 single end analysis. And as museum specimens has to have the DNA degraded through time, we also perform a evaluation of the DNA degradation. So we use mapped image that identifies depurination and deamination, and to perform the rest of the analysis. So we also align the reads against the Ceratina coccolata genome, and we filter for low quality data. For the genetic diversity analysis, we used the population twin stacks and the genodive. For the population structure, we use admixture and PCA plots. And we also search for candidate genes. So we search for outlier loci using PCADEPT and search for candidate genes. And also we saw which goals were enriched using TopGo. For the climate analysis, we separated the in 30 years bands for historic and contemporary. So for historic data, we analyzed climatic data from 1958 to 1988. And for the contemporary, we analyzed data from 1989 to 2019. And we gathered data from mean, maximum, and minimum temperature, and also for precipitation. And for land use change, we used satellite images from 1972 and from 2018. And we classified the images according to 6 land use classes: bare soil, forest, agriculture or pasture, and scrub, urban, and water. So comparing those two classified images, we saw that we had, from the sample spots, 4 land use classes increasing and 2 decreasing. We had increasing bare soil, forest, urban areas, and water, and decreasing, we have agricultural pasture and scrubland. For the climate data that we analyzed, we found significantly increasing in mean and minimum temperature. And how the bees are being affected genetically for these kinds of change. So the genetic diversity is actually decreasing through time. So you can see that the ancestral population from 1968, the heterozygosity index is actually increasing. So as you can see from the HS. And the inbreeding coefficient, which is measure as the FIS here, 0.066, is actually increasing. So this means that these bees are losing genetic diversity and are becoming more inbred. This is true for Ceratina cocurata, and this is also true for Ceratina dupla, that have the same pattern of decreasing in genetic diversity and increasing in FIS, the breeding coefficient. For the population structure analysis, we have two genetic clusters that we can see clearly. So you can see from K = 2 that you have from the admixture plots from your left, you can see 2 genetic clusters. And from the PCA results, the PCA plots, you also can have the historic in red and contemporary in blue, forming 2 genetic different clusters for Ceratina cocurata and also for Ceratina dupla, where you can see the admixture plots on the left and the PCA plots on your right. As for the candidate genes, for C. cocurata, we found 892 outlier SNPs that yielded 270 genes and enriched 89 GO terms. Among specific genes that were candidate genes that were found here, we find cytochrome P450 family that we could see from the past talks here that are responsible for immune response, and also is related to insecticide tolerance in Hymenoptera. We also find candidate genes related to G protein coupled receptors that are responsible for toxicological responses and also insecticide resistance. And also candidate genes related to glutamate receptor, which are related to learning and memory mechanisms. We also found GOs associated with muscle development. This for C. cocurata. For C. dupla, we found 794 outlier SNPs, and they yielded 225 genes and reached 50 GO terms. And among genes, among candidate genes for C. dupla, we found ecdysone receptor, which are related to ecdysis and post-ecdysis process, and also related to resistance to heat, to starvation, and to oxidative stress. And we found GO terms related to viral responses and calcium-dependent activity. So what all these results means for these bees? So Ceratina cocurata and Ceratina dupla are losing suitable habitat, as we saw that scrubland area is becoming smaller. And this is crucial for these bees because they're highly reliant on these small trees and scrubs to provision pollen and for nesting. And also urbanization is increasing. So they also are losing suitable habitat. And also water and bare soil is decreasing. And this is affecting these bees populations in terms of increasing in breeding, as we could see by the FIS, and also the lower genetic diversity through time. And they also showing the higher genetic structure as we saw with different clusters from ancestral and contemporary populations. But also, they showing signatures of selection to insecticide resistance and to immune response. And this is very interesting because it's very relatable to how increasing agriculture and urbanization is increasing uses of pesticides. And importantly, exposure to pesticide has been reported as a crucial issue regarding bees' health and population decline during the past decades. And they also are showing signatures of selection to resistance to heat and to muscle development, according here as flight muscle. And this is also important because the planet is getting warmer and urbanization is increasing and cities also are getting warmer due to heat island effect. So our results offer insights into the selective stressors that these bees have to handling, and also to abiotic pressures underlying these populations. And also it's very important to understand how this is all affecting these bees, because improves conservation planning for those, specifically for urbanization process. And also the museomics can also be seen as an era of discovery, in that the questions we can ask are infinite. And I would like to thank the Rehan Lab, Rain Lab, and the Royal Ontario Museum, and all the audience and everybody that is watching. Thank you. I'm sorry, I didn't. 1989. Oh, from the climate data. Yeah, we tried to put 30 years of a scale against 30 years, just to be sure that we weren't getting any kind of, to be sure that that is climate relatable, that is not just a confluence of year from year. So yeah, so we did an average from each year, and then compare those two. And those significantly different just for mean temperature and minimum temperature that are increasing significantly. The other ones wasn't significantly. Yeah. Yes. So how do we get pollen DNA? Oh yeah, we had this quantification of how good the DNA was, and was usable. And then we have, because the paper is not out yet, but we have all this. And if you have, I can. So if you need, I can talk to you also after. But it's a good quality. We just had to take out the DNA that was highly degraded from germination and depurination, and otherwise it's very was useful. The quality. Yes. Hi. Thank you. Yeah, so we ran this population structure analysis against a lot of different softwares, and we just saw the K2 was better. Yeah, and this was run from two populations only, like for ancestral and contemporary for each species and separate. Yeah, they're not quite totally a admixture, yeah, but the K2 was the best for the results. Again, yeah. But thanks for asking.